Good day, everyone. My name is Daryl A. Gregory, and my assigned topic to me is all about cell and molecular biology. So what is cell and molecular biology? It is comparative study of structure and function of organisms and biological processes at the level of cells and the macromolecules that define them, such as DNA, RNA, proteins, lipids, and carbohydrates. These are the following topics in cell and molecular biology that we are going to discuss. First is the cellular basis of life, the type of cell, cell structure, chromosomal structure and genes, cell cycle, DNA replication, DNA transcription, membrane-bound organelles, the cytoskeleton and cell motility, and the cellular metabolism. Before we're going to start our review proper, let's gonna talk about the cell. So what is cell? Cell are the basic building blocks of living things. We human, our body is composed of trillions of cells, because the cells are the basic structure of living things. And also, cells provide structure for the body, take in nutrients from food, and carry out important functions. Cells group together to form tissue, which in turn group together to form organs, such as heart and brain. Our cells contains a number of functional structures called organelles. These organelles carry out tasks such as making proteins, processing chemicals, and generating energy for the cell. These are the following proponents of cell and molecular biology. First is Robert Hooke. The cell was first discovered by English scientist Robert Hooke in 1665, which can be found to be described in his book Micrographia. One of his observations was form very thin slices of bottle cork. Hooke discovered a multitude of tiny pores that he named cell. The Latin word cilia, meaning a small room like monks live in, and also cellulae, which he described the cork as made up of hundreds of little hexagonal boxes, giving a kind of honeycomb appearance. However, Hooke did not know their real structure or function. He had, though, the cells were actually empty cell walls of plant tissue, and mentioned the Greek prefix cytos, means hollow vessel. With microscope during this time having a low magnification, Hooke was unable to see that there were other internal components to the cell he was observing. His cell observation gave no indication of the nucleus and other organelles found in most living cells. Hence, Robert Hooke studied and discovered the cell from a thin slice of cork in the bark of oak tree. But that was a dead cell. Robert Hooke saw only a dead walls of plants. These cells had lost their living contents and looked like small rooms. Second is Anton van Leeuwenhoek. First discovered a free-living algae, Spirogyra cells in water in the pond in 1674. With the improvement of microscope, the living cells were first discovered by Anton van Leeuwenhoek. He observed living cells and called them animalcules. Some small animalcules are now called bacteria. The invention of the microscope supported the study of finer details of a living cell. Next, Theodor Schwann and Matthias Leiden. These two scientists proposed a cell theory, which is the cell is the unit of structure, physiology, and organization in living things. The cell retains a dual existence as a distinct entity and a building block in a construction of organisms. Cells are by free cell formation similar to the formation of crystal or the spontaneous generation. Next is Rudolf Virchow. We know today that the first two tenets are correct, but the third is clearly wrong. The correct interpretation of cell formation by division was finally promoted by others and formally enunciated in Rudolf Virchow's powerful dictum called the Omnis Cellula e Cellula, also known as all cells only arise from pre-existing cells. Phoebus 11 identified the components, the four bases, the sugar and the phosphate chain, and he was showed that the components of DNA were linked in a order phosphate-sugar base. And he also analyzed the chemical properties of nucleic acid. He discovered that DNA was abundant in three things: 5-carbon sugar or the pentoses, the phosphate, as a mixture had previously found, and nitrogen bases. James Watson and Francis Crick discovered the double helix structure of DNA molecule. In an influential presentation, Crick laid out the central dogma which foretold the relationship between DNA, RNA, and proteins and articulated the sequence hypothesis. George Biddle and Edward Tatum demonstrated the existence of a precise relationship between genes and proteins. Through experiments on the red bread mold or Neurospora crassa showed that genes act by regulating distinct chemical events, affirming the one gene, one enzyme hypothesis. They concluded that the function of a gene is to direct the formation of a particular enzyme, which regulates a chemical event. A mutation can alter a gene so it no longer produces the normal enzyme, resulting in a physical symptom, like a need for nutritional supplements. Biddle and Tatum proposed that, in general, each gene directs the foundation of one, and only one, enzyme. Oswald Avery demonstrated that genes are made up of DNA. A team of scientists led by Oswald Avery at the Rockefeller Institute rigorously followed up on this experiment in 1940s. They found that a pure extract of transforming principle was unaffected by treatment with protein-digesting enzyme but was destroyed by a DNA-digesting enzyme. This showed that the transforming principle is DNA, and by extension, a gene is made of DNA. Still, many scientists were slow to accept this clear proof that DNA, not protein, is the genetic molecule. Richard Altman. He first described the mitochondria. He called them bioblasts and concluded that they were elementary organisms living inside cells and carrying out vital function. Altman would have been greatly satisfied by knowing that his idea of the symbiotic origin of mitochondria would be revived several decades later, based on the similarities between mitochondria and bacteria. Walter Fleming. He described chromosome behavior during mitosis. He also analyzed that structure and processes in the cell nucleus under a microscope. He was the first person to conduct a systematic study of chromosomes during division and called this process mitosis. Camillo Golgi described the Golgi apparatus. He described in nerve cells an internal reticular apparatus impregnated by a variant of his chromoargentic staining. Bert Gorter and Francis Grendel described the basic structure of the plasma membrane. They found that when the lipids spread out as one layer, the area that they covered was almost exactly twice the surface of the red blood cells themselves. Thus, Gorter and Grendel concluded that the lipid surface surrounding the cell must be two layers. The cellular basis of life. Cell as the fundamental unit of life. A cell is a small, membrane-enclosed unit filled with a concentrated aqueous solution of chemicals and endowed with the extraordinary ability to create copies of themselves by growing and then dividing into two. The simplest forms of life are solitary cells. All living things are groups of cells derived by growth and division from a single building block of founder cell. Every animal or plant is an enormous collection of individual cells, each of which executes a specialized role that is combined by complex systems of cell-to-cell communication. Indeed, cell is the fundamental unit of life. Living things may be unicellular like bacteria or multicellular like animals, plants, and fungi. Cell structure is diverse, but all cells share common characteristics. So let's talk about the cell theory. The cell theory came about through the painstaking efforts of the scientists during the 17th century. Discovered by Schwann and Schleiden and Virchow. The cell theory states that all organisms are composed of one or more cells. The cell is a structural unit of life, and all cells exist from pre-existing cells. Basic properties of cell. The first basic properties of cell is cells are highly complex and organized. Cells arises with complexity in structure, organization, and function. Through complicated but each structure interplay with high precision in carrying out cellular activities efficiently. For example, during DNA duplication, the occurrence of an error is less than one mistake for every 10 million nucleotides incorporated, greatest of which are rapidly corrected via elaborate repair mechanism that identifies the defect. Second basis properties of cells are the cells possess a genetic program and the means to use to it. Cells have DNA and organisms are built according to the information encoded in a collection of genes. These genes are packaged into a set of chromosomes that occupies the space of a cell nucleus. Genes are more than storage lockers for information. They constitute the blueprints for constructing cellular activities and the program for making more of themselves. Cells are capable of producing more themselves. Cells are reproduced by division. It is a process in which the content of a mother cell are distributed into two daughter cells. Cells are acquired and utilize energy. Every biological process requires the input of energy. The energy from sunlight is trapped by light-absorbing pigment and converted into chemical energy through photosynthesis. This chemical energy is then stored in plants in the form of carbohydrates. For most animals, sugar glucose is released by the liver. The stored energy in glucose will then be broken down by respiration process to produce energy for movement and other related physiological activities. Next is cells carry out a variety of function. Cells are likely miniaturized chemical plants. They perform a number of metabolic activities to maintain homeostasis. Virtually, all chemical changes that take place in cell require enzymes, molecules that greatly increase the rate at which a chemical reaction occurs. The sum total of the chemical reaction in a cell represents the cell metabolism. Next is cell engaged in mechanical activities. Cells are sites of bursting activity. Materials are transported from place to place, structures are assembled and then rapidly disassembled. And in many cases, the entire cell moves itself from one site to another. These activities are based on dynamic mechanical changes within cells, many of which are initiated by changes in the shape of motor proteins. Motor proteins are just one of many types of molecular machines employed by cells to carry out the mechanical activities. Next is cells are able to respond to stimuli. Some cells respond to stimuli in obvious ways. For example, a single-celled protist moves away from an object in its path or moves toward a source of nutrients. Cells within a multicellular plant or animal respond to stimuli less obviously. Most cells are covered with receptors that interact with substances in the environment in highly specific ways. Cells possess receptors to hormones, growth factors, extracellular materials, and substances on the surface of the cell. A cell's receptors provide pathways through which external stimuli can invoke specific responses in target cells. Cells may respond to specific stimuli by altering their metabolic activities, moving from one place to another, or even committing suicide. Next is cells are capable of self-regulation. The importance of cells' regulatory mechanism becomes most evident when they break down. For example, failure of a cell to correct a mistake when it duplicates its DNA may result in a debilitating mutation or a breakdown in a cell's growth control safeguard which enable it to transform into a cancer cell with a capability of destroying the entire organism. The last is the cells evolve. It is presumed that cells evolved from some types of pre-cellular life form, which in turn evolved from non-living organic materials that were present in the primordial seas. All living organisms have evolved from a single, common ancestral cell that lived more than 3 billion years ago. The two types of cell. Fundamentally, there are two types of cells, the prokaryotic and the eukaryotic cells. These two cell types were classified based on the presence or absence of a nucleus. Organisms whose cells have a nucleus are called eukaryotes, from the Greek word eu meaning well or truly, and karyon, a kernel or nucleus. Organisms whose cells do not have a nucleus are called prokaryotes, from pro meaning before. First, let's talk about eukaryotic cells or the eukaryotes. All eukaryotic cells have a nucleus, but possession of a nucleus goes hand in hand with possession of a variety of other organelles, most of which are membrane enclosed and common to all eukaryotic organisms. Eukaryotic organisms include plants, animals, fungi, and protists. Eukaryotic cells can be unicellular or multicellular. The other types of cell is the prokaryotic cells or the prokaryotes. defined as those that do not possess a true nucleus or membrane-bound organelles. The distinction between prokaryotic cell and the eukaryotic cells is based on the structural complexity rather than phylogenetic relationship. There are two types of prokaryotic cells or prokaryotes, which is archaea and bacteria. Archaea, they are prokaryotic unicellular organisms that reproduce asexually. They include several groups of organisms whose evolutionary ties to one another are revealed by similarities in the nucleotide sequences of their nucleic acid. They live in all sorts of environments, but they are known for thriving in extreme environments, though to be similar to those of early Earth. For example, the methanogens, which live in environments like swamps and marshes and the guts of animals. The halophiles are salt lovers living in the bodies of water, such as the Great Salt Lake in Utah. The thermophiles are both lovers of high temperature and acid. Bacteria. This domain includes the smallest known cells, the Mycoplasma, which is measured 0.2 diameter, which are only known prokaryotes to lack a cell wall and to contain a genome with fewer than 500 genes. They are also diversified and plentiful and are found in large number nearly everywhere on Earth. This is an example of eukaryotic cell and the different organelles inside the cell, as well as the prokaryotic cell and its parts. I believe we already know this topic already, right? As far as I know, it was already discussed back in high school or even in college in our genetic class. So now, let's refresh our mind with the different organelles and its function. Cell structure. Nucleus. Stores genetic information in extraordinarily long DNA molecules called chromosomes. Nuclear envelope. A double membrane that separates the nucleus from the cytoplasm. Nuclear pore. It is where all traffic passes into and out of the nucleus. Nucleolus. Assemble ribosome from more than 50 different proteins and 3 RNA molecules. Chromatin. DNA plus the protein that package it within the cell nucleus contains the genetic information. Cytoskeleton maintains the shape of most cells. Centrioles may be involved in microtubule formation and disassembly during cell division and in the organization of cilia and flagella. Mitochondrion, powerhouse of the cell. Its enzymes use most of the energy released from the breakdown of nutrients to synthesize ATP, the common currency of most energy requiring reaction in cell. Golgi apparatus or Golgi body processes the sugar side chain on transmembrane and secreted proteins. It consists of a stock of flattened, membrane-bound sacs with many associated vesicles. Endoplasmic reticulum, a continuous system of flattened membrane sacs and tubules. Endoplasmic reticulum has two types, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough ER, studied with the ribosomes and is the site of the protein synthesis and processing. Smooth ER, lacks ribosomes and is the site of synthesis of phospholipids and the packaging of proteins into vesicles, among other functions. Lysosome, are small, irregular-shaped organelles in which intracellular digestion occurs, releasing nutrients from ingested food particles into the cytosol and breaking down unwanted molecules for either recycling within the cell or excretion from the cell. Cytosol, a semi-fluid medium that is the site of many chemical reactions that are fundamental to the cell existence. Plasma membrane regulates what enter and exit the cell. Ribosome catalyze the synthesis of proteins using the nucleotide sequence of mRNA molecules to specify the sequence of amino acids. Ribosome free in the cytoplasm synthesize proteins that are released for routing to various intracellular destinations. Chloroplast capture energy from sunlight site for photosynthesis. Cell wall provides protection and rigidity to the cell. Vacuole stores a variety of substances for the cell. Anything from water to plant pigments and toxic substances are stored in these membranous sacs. Helps regulate turgor pressure of the plant cell. Peroxisomes are membrane-bound organelles containing enzymes that participate in oxidative reaction like mitochondria. Peroximal enzyme oxidize fatty acids, but the energy is not used to synthesize ATP. Now, let's move to the chromosomal structure and genes. The nucleus. The nucleus of the cell is the brain of the cell. It controls the entire activity of the cell. Its structure is very much complex and protected by a nuclear envelope. The molecules pass through a highly organized nuclear pore. It also has a nucleolus where ribosome subunits are synthesized. Chromatins, which are thread-like structure condensed to form chromosomes during cell division, are confined and suspended in the nucleoplasm. The two essential central cellular processes encompassing replication and transcription occur inside it. The regulation in the expression of genes among eukaryotic organisms is also governed by this organelle. The nucleus has the following key features including the outer membrane, inner membrane, nuclear pore, and nucleolus. The nuclear envelope has a double membrane and encloses the contents of the nucleus. The outer nuclear membrane is continuous with the rough endoplasmic reticulum. The nuclear pore, as you have learned previously, is the opening where all traffic passes into and out of the nucleus. The nuclear pore is a complex structure. It is one of the largest protein complexes in the cell. It is easily distinguishable using scanning electron microscopy. Chromosome. It is a huge DNA molecule which can be proliferated steadily by innumerable groups of dividing cells. Chromosomes are thread-like structure present in the nucleus, which carries genetic information from one generation to another. They play a vital role in cell division, heredity, variation, mutation, repair, and regeneration. A DNA molecule with its attendant proteins that moves as an independent unit during mitosis and meiosis. Before DNA replication, each chromosome consists of a single DNA molecule plus protein and is called a chromatid. After replication, each chromosome consists of two identical DNA molecules plus proteins. They are called sister chromatids. Chromosomal DNA molecules are usually linear, but can be circular in organelles, bacteria, and viruses. The chromosome is consisting of chromosomal locus that controls the movement of chromosomes during cell division. The centromere is the certain DNA sequences which have protein that attach to them, albeit epigenetic factors also function as major parts. Among higher eukaryotes, this structure can be viewed as a constricted region whereby sister chromatids are linked together mostly strictly. The portion of the chromosomes that are not in the centromere itself are called chromosome arms. Another specialized structure found in chromosome is the telomere. It can be found at either end of the chromosome DNA molecules that endorse the complete replication of the chromosomal ends and protects the end within the cell. However, this structure is not found among most prokaryotes and in mitochondrial DNA because they are secular in form. Naturally occurring eukaryotic nuclear chromosomes are generally on linear DNA molecule that stretches between the telomere at either end. There are classes of chromosomes. They are classified based on the location of centromere relative to the arms. In metacentric chromosome, the centromere is located midway along the chromatid. In submetacentric chromosome, the centromere is located as asymmetrically so that each chromatid can be divided in short and long arms. In acrocentric chromosome, the centromere is located near the end of the arms. In telocentric chromosomes, the centromere appears to be located at very near the end of the chromatid. Gene. Chromosomes are carrier of genes. A gene is a segment of DNA that codes for RNA or a set of proteins. It is a unit of hereditary and it controls the hereditary characteristics of an organism, which are transmitted from generation to generation. Structure of prokaryotic gene. A bacterial gene is composed of a single stretch of uninterrupted nucleotide sequence which encodes the amino acid sequence of a protein. The DNA of prokaryotes is organized into a circular chromosome supercoiled in the nucleoid region of the cell cytoplasm. Proteins that are needed for the specific function are encoded together in the block called operons. For example, all of the genes needed to use lactose as an energy source are coded next to each other in the lactose or lac operon. In prokaryotic cells, there are three types of regulatory molecules that can affect the expression of operons, repressor, activators, and inducers. When we say repressors, are proteins that suppress transcription of gene in response to an external stimulus, whereas activators are proteins that increase the transcription of a gene in response to an external stimulus. Finally, inducers are small molecules that either activate or repress transcription depending on the needs of the cell and the availability of substrate. In bacteria and archaea, structural protein with related functions such as the genes that encode the enzymes that catalyze the main steps in a single biochemical pathway are usually encoded together within the genome in the block called an operon, and are transcribed together under the control of a single promoter. This form of polycystonic transcript, the promoter then has simultaneous control over the regulation of the transcription of these structural genes because they will either all be needed at the same time or none will be needed. In prokaryotes, structural genes of related function are often organized together on the genome and transcribed together under the control of a single promoter. The operant regulatory region includes both the promoter and the operator. If a repressor binds to the operator, then the structural genes will be not transcribed. Alternatively, activators may bind to the regulatory region, enhancing transcription. Structure of eukaryotic gene. A eukaryotic gene commonly consists of coding sequences called exons and non-coding or the intervening sequences called introns. Both sequences are transcribed to produce long RNA transcripts. However, introns are spliced to produce a mature RNA. Although introns are the intervening sequence, they too play a few functions. Some introns encode functional products, for example, proteins and RNAs. Some also help in the regulation of gene expression, which is very important to the cell performance as they contain regulatory sequence. Deregulatory sequence also allow alternative splicing. So now, let's move to the cell cycle and DNA replication. Cell cycle. The cell cycle is the sequence of events that leads to the duplication and division of a cell. Cell cycle functions for the accurate duplication of the vast amount of DNA in the chromosomes and then to segregate the DNA into genetically identical daughter cells such as each cell receive a complete copy of the entire genome. The eukaryotic cell cycle generally ensues in 4 phases. The cell grows continuously during interphase, which consists of 3 phases, G1, S, and G2. DNA replication is confined to S phase. G1 is the gap between M phase and S phase, and G2 is the gap between S phase and M phase. During M phase, the nucleus divides in a process called mitosis. Then the cytoplasm divides in the process called cytokinesis. In G1 phase is naturally the longest and the utmost flexible phase of the cell cycle. When the cells are born during cytoplasmic division, they are roughly half the size they were before mitosis. It is during G1 phase where they grow back toward an optimal size. During this period, several events involved in the cell cycle progression are curbed in order to the cell to not initiate a new round of production. This repressive control system is known as the restriction point. If the condition including the supply of nutrients is insufficient, such stimulus may signal to start on terminal differentiation, causing the delay of the progress through the cell cycle in G1, or exit the cycle and enter G0. However, if the conditions are right or positive stimuli are received, cells overcome the restriction point block and activate a program of gene expression that commits them to a new cycle of DNA replication and cell division. Cancer cells often have faults in restriction point control of remaining to grow and go to the divide in the absence of appropriate environmental signals. G0 and growth control. Not all multicellular organisms proceed to cell division, as most of them will differentiate to carry out their specialized function. These cells, in the G0 phase, most often cells enter G0 directly as they exit their previous mitosis. G0 cells are not resting and they are often actively engaged in the synthesis of protein and secretion, and they may also highly active. The G0 cells are not necessarily permanent, as some of them are also conscripted to go on the cell cycle in response to the certain stimuli. This process requires to be highly controlled, and the unregulated production of cells in multicellular organisms may lead to cancer. In S phase, this phase is a relatively short period during which major enzymatic events that will activate the entry to mitosis progressively accumulate and are transformed to active forms. If such event reach a critical threshold level, the cell enters mitosis. During this period, the chromatin and cytoskeleton are redyed for the structural transformation that may happen during mitosis. Once there is damage in the DNA and is detected during this phase, the checkpoint activates the DNA damage response and suspends the entry of the cell into the next phase to the cycle, which is mitosis. The M phase. This phase involves mitosis and the subsequent cytokinesis, whereby chromosomes and cytoplasm are divided into two daughter cells. Mitosis is generally divided into 5 distinct phases, including prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the one set of chromosome condensation takes place. Take apart inside the nucleus and the mitotic chromosomes start to form a typical arrangement of loops. In the cytoplasm, a dramatic transformation in the dynamic properties of the microtubules decrease their half-lives from approximately 10 minutes to approximately 30 seconds. The duplicate centromere separates and form the two poles of the mitotic spindle. During prometaphase, this phase starts with the nuclear envelope breaks down, especially among the eukaryotes. Chromosome starts to attach haphazardly to microtubules coming from the two poles of the forming mitotic spindle. Other microtubules emanate on chromosomes and within the mitotic spindle as both the kinetochores on a pair of sister chromatids attached to microtubules form opposite spindle poles. The pairs of chromatids slowly moves to a point midway between the poles. During metaphase, the chromosomes are already reached the midway and aligned at the center. During anaphase, sister chromatids are separated and move to the spindle poles wherein they move toward the opposite spindle poles. Telophase. During this phase, the nuclear envelope reforms on the surface of the chromatin. A contractile ring of actin and myosin then assembles as a circumferential belt at the cortex halfway between spindle poles and compresses the equator of the cell. The last is the cytokinesis. Follows telophase as a cytoplasmic division occurs, separating daughter cells from one another. We all know that there are two types of cell division. It is mitosis and meiosis. So mitosis is what I have mentioned earlier. So now let's talk about meiosis. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction with the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring. Meiosis I. The first meiotic division begins with prophase I. During prophase I, the complex of DNA and protein known as chromatin condenses to form chromosomes. The pairs of replicated chromosomes are known as sister chromatids, and they remain joined at the central point called the centromere. A large structure called the meiotic spindle also form along proteins called microtubules on each side or pole of the cell. Between prophase 1 and metaphase 1, the pairs of homologous chromosomes form tetrads. Within the tetrad, any pair of chromatid arms can overlap and fuse in the process called crossing over or recombination. Recombination is the process that breaks, recombines, and rejoins sections of DNA to produce new combination of genes. In metaphase 1, the homologous pairs of chromosomes align on either side of the equatorial plate. Then in anaphase 1, the spindle fiber construct and pull the homologous pairs, each with two chromatids, away from each other and toward each pole of the cell. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes the process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. Each daughter cell is a haploid and has only one set of chromosomes, or half the total number of chromosomes of the original cell, and undergo the meiosis II. Is a mitotic division of each of the haploid cells produced in meiosis I. During prophase II, the chromosome condense, a new set of spindle fibers forms. The chromosomes begin moving towards the equator of the cell. During metaphase II, the centromeres of the paired chromatids align along the equatorial plate in both cells. Then in anaphase II, the chromosome separates at the centromeres. The spindle fibers pull the separated chromosomes toward each pole of the cell. Finally, during telophase II, the chromosomes are enclosed in nuclear membranes. Cytokinesis follows, dividing the cytoplasm of the two cells. At the conclusion of meiosis, there are 4 haploid daughter cells that go on to develop into their sperm and egg cells. DNA replication. DNA replication is the process of copying DNA which takes place inside the nucleus. It involves complementary base pairing, allowing each strand to serve as a template for a new strand. The process is also called as a semi-conservative given that the new strand is formed from the old template strand. The process of DNA replication are initiation, primer synthesis, leading strand synthesis, lagging strand synthesis, primer removal, ligation, and determination. For the initiation, replication begins at a specific site termed as the origin of replication, which has a specific sequence of nucleotides which can be recognized by the initiator proteins. These proteins bind to this site, thus flagging it for the docking of other proteins and enzymes. An enzyme helicase is recruited to the site to unwind the double helix. DNA helicase breaks the hydrogen bonds between the bases, producing 2 single strands. This point or region is known as the replication fork. The separated DNA strand naturally wants to reform, stabilizing the hydrogen bond. A special binding protein binds each of the single strands to prevent them from reassociating. The DNA replication process thus initiates, and the replication fork proceeds into 2 opposite directions. Next is the primer synthesis. The synthesis of complementary DNA strand from the existing DNA strand is brought about the DNA polymerase. However, DNA polymerase cannot add new nucleotides in the independent manner that requires 3' end hydroxyl group. DNA primase provides the primer, and this serves the platform of the DNA polymerase to extend new nucleotides and synthesize a complementary strand. However, the replication process proceeds. DNA supercoil, making it difficult for the DNA strand to be unwound. Thus, special protein bind to the DNA strand ahead of the replication fork. Next is the leading strand synthesis. DNA polymerase can read the DNA strand in the 3' end to the 5' end direction, and thus it can synthesize new complementary strand in the 5' end to 3' end direction. Once primer is provided, DNA polymerase extend new nucleotides from 3' end OH end of the primer. However, DNA strands run in opposite directions. Thus, in one strand, DNA polymerase synthesizes the new DNA strand in a continuous manner. This is the leading strand. A sliding clamp binds the DNA to keep it in place. Lagging strand synthesis. In an opposite DNA strand, DNA polymerase synthesizes new complementary strand in a back-stitching mechanism. DNA primase provides a short RNA primer at regular interval. Thus, DNA polymerase synthesizes the new strand piece by piece. This is called the Okazaki fragments, which are later joined to form a chain of nucleotides. This is the lagging strand since the process occurs in a slow rate. Primer removal. Although new strand has already been synthesized, the primer present on it requires to be removed. A special protein specifically removes the primer in the 5' end to 3' end direction via polymerase exonuclease activity, and then replace it with deoxyribonucleotide via the 5' end to 3' end DNA polymerase activity. Next is ligation. In the lagging strand, nicks and gaps are still present between the adjacent Okazaki fragments. An enzyme ligase identifies the seals of these gaps by creating a phosphodiester bond between the 5' end phosphate and the 3' end OH groups of Okazaki fragments. The last process of DNA replication is the termination. The DNA replication stops when it encounters a terminator. It is a special sequence of nucleotides that signals the end of replication. A special protein will bind the terminator sequence physically, blocking the helicase. Thus, when helicase encounters this protein, it falls off along with her other binding proteins. DNA transcription. Transcription is the process which uses DNA as a template to synthesize RNA. Like DNA, it also occurs in the nucleus of the cell. Mechanism of transcription: initiation, elongation, and termination. In initiation process, DNA transcription begins when a group of proteins called transcription factors bind to a special sequence of nucleotide called the promoter. It signals RNA polymerase II to join the complex, forming the initiation complex, a transcription factor. The TF2H then begin the unwinding of the DNA at the transcription start site, making the RNA polymerase to escape from the promoter region and begin to break the hydrogen bond between the bases. RNA polymerase then proceed the transcription process, leaving the promoter region. Next process is the elongation. As the RNA polymerase unwind the double helix, a transcription bubble forms, exposing the two single strands of DNA. Only one of these strands is used as a template. Like DNA polymerase, RNA polymerase can only synthesize a new DNA strand from 3' end to 5' end direction, and thus it synthesizes RNA transcript from 5' end to 3' end direction. Ribonucleotide triphosphate from the cytoplasm are paired up on the complementary bases of the exposed DNA template. RNA polymerase joins the ribonucleoside triphosphate to synthesize the pre-mRNA. As the RNA polymerase advances, the process continues. The DNA strand that has been transcribed rewinds and forms the double helix. The last process is the termination. Transcription stops when RNA polymerase, a special nucleotide sequence that signals the end of transcription. RNA polymerase and the pre-mRNA transcript detach from the DNA. The termination sequence usually consists of a series of adjacent adenines preceded by a nucleotide palindrome that gives an RNA molecule that assumes a stem and loop configuration. This configuration stops RNA polymerase from transcribing any further. Now, let's move to the membrane-bound organelles. The first organelle is the endoplasmic reticulum. Endoplasmic reticulum, or ER, is an organelle that consists of a system of membranes. Enclosed within it is a wide space called lumen that is isolated from surrounding cytosol through the ER membrane. It has highly dynamic configuration and it is undertaking constant turnover and restructuring. The ER is divided into two subcompartments, the rough endoplasmic reticulum, or the RER, and the soft endoplasmic reticulum, or the SER. The rough endoplasmic reticulum is redefined because of the presence of ribosomes attached to its cytosolic surface, while smooth endoplasmic reticulum lacks bound ribosome. The rough endoplasmic reticulum is typically composed of a network of flattened sacs, or the cristernae, where each layer is connected to its neighbors by helicoidal membrane. The rough endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope, which also bears ribosome on its cytosolic surface. In contrast, the membrane of the smooth endoplasmic reticulum are highly curved and tubular, forming an interconnecting system of pipeline traversing the cytoplasm. When cells are homogenized, the smooth endoplasmic reticulum tubules fragment into smooth surface vesicles, whereas the rough endoplasmic reticulum sheets fragment into rough surface vesicle. So, other function of endoplasmic reticulum. First is the smooth endoplasmic reticulum. It synthesizes the steroid hormones in the endocrine cells of the gonad and adrenal cortex, and detoxification in the liver of the wide variety of organic compounds, including barbiturates and ethanol, whose chronic use can lead to the proliferation of the smooth endoplasmic reticulum in liver cells. Another, it is sequestering calcium ions with the cytoplasm of cells. The regulated release of the calcium, or the Ca2+, from the smooth endoplasmic reticulum of skeletal and cardiac muscle cells, known as the sarcoplasmic reticulum in the muscle cells, that triggers contraction. Next is rough ER, or rough endoplasmic reticulum. It synthesizes proteins and synthesis of the most lipids of cell membrane. Golgi apparatus. The Golgi complex, or the Golgi apparatus, has the characteristic morphology consisting primarily of flattened disc-like membranous cristae with dilated rims and associated vesicles and tubules. The Golgi complex is divided into several functionally distinct compartments arranged along an axis from the sys, or entry phase closest to the endoplasmic reticulum, to the trans, or exit phase at the opposite end of the stack. The cis-most face of the organelle is composed of an interconnected network of tubules referred to as the cis-Golgi network, or the CGN. The CGN is thought to function primarily as a sorting station that distinguishes between protein to be shipped back to the ER and those that can proceed to the next Golgi station. The bulk of the Golgi complex consists of a series of large flattened cristae which are divided into cis, medial, and trans cristae. The transmost face of the organelle contains a distinct network of tubules and vesicles called the trans-Golgi network, or the TGN. The TGN is a sorting station where proteins are segregated into different types of vesicles heading either to the plasma membrane or to various intracellular destinations. The membranous elements of the Golgi complex are, though, to be supported mechanically by a peripheral membrane skeleton or scaffold composed of a variety of proteins, including members of the spectrin, ankyrin, and actin families, proteins that are also present as part of the plasma membrane skeleton. The Golgi scaffold may be physically linked with motor proteins that direct the movement of vesicles and tubules entering and exiting the Golgi complex. A separate group of fibrous proteins are thought to form a Golgi matrix, which plays a key role in disassembly and reassembly of the Golgi complex during mitosis. So other function of Golgi apparatus. It serves as a processing plant as a newly synthesized membrane protein and secretory of lysosomal proteins, leave the endoplasmic reticulum and enter to the Golgi complex at its cis phase and then pass across the stack to the trans phase along the stack proteins that were originally synthesized in the rough endoplasmic reticulum are sequentially modified in specific ways. Also, the site of synthesis of most of the cell complex polysaccharides, including glycosaminoglycan chains of the proteoglycan and the pectins and hemicellulose found in the cell walls of plants. The lysosome. Lysosomes are an animal cell's digestive organelles. A typical lysosome contains at least 50 different hydrolytic enzymes produced in the rough ER and targeted to these organelles. Taken together, lysosomal enzymes can be hydrolyzed virtually every type of biological macromolecules. The enzymes of a lysosome share an important property. All have their optimal activity at an acid and thus are acid hydrolases. The pH optimum of these enzymes is suited to the low pH of the lysosomal compartments, which is approximately 4.6. The high internal proton concentration is maintained by a proton pump present in the organelle's boundary membrane. Lysosomal membrane also contain a variety of high glycosylated integral proteins whose carbohydrate chains are thought to form a protective lining that shields the membrane from attack by enclosed enzymes. The function of lysosomes are it breaks down the materials brought in the cell from the extracellular environment. It also plays a key role in organelle turnover, that is, the regulated destruction of the cell's own organelles and their placement. The peroxisome. Peroxisomes are another kind of microbodies that are single membrane like lysosomes. It contains oxidases and catalyses, responsible for degrading fatty acids, and also it is responsible for degrading toxic compounds. Now, let's move to the cytoskeleton and cell motility. Microtubule. Microtubules are rigid, rod-like polymers of tubulin, a single type of globular protein that give assistance for a diversity of cellular assemblies and traces for activities driven by motor proteins known as kinesins and dyneins. The structure of microtubules, dimer of alpha and beta tubulin polymerize to form microtubules, which are composed of 13 protofilaments arranged around the hollow core. These tubulate dimers polymerized to form microtubules are made up of 13 linear protofilaments that are brought together around hollow core. Polymerization is the process in which relatively small molecules called monomer combine to generate a huge network of molecules called a polymer. These protofilaments, which are made up of head-to-tail arrangement of tubulin dimers, are arranged in parallel pattern. Accordingly, microtubules are polar assemblies which consist of two dissimilar ends, the end with alpha tubulin showing it called its plus end, and the opposite end which contains exposed alpha tubulin is called the minus end. Such polarity defines the direction of molecular motion by microtubule. So what is the function of microtubule? As one of the principal components of the cytoskeleton, microtubules function for defining cell shape and joining in several cell movements, encompassing several forms of cell locomotion, the intracellular transport of organelles, as well as the separation of chromosomes during cell division. Specifically, microtubules are involved in the beating of cilia and flagella. Microtubules are organized in the centrosome of animal cells. Centrosomes are absent in plants. Centrosomes serve as the organizing centers that regulate the location, number, and orientation of the microtubules. The centrosome has a pair of centrioles that is covered by matrix of proteins. The centrosome matrix includes hundreds of ring-shaped structures designed from a special type of tubulin called white tubulin, and each white tubulin ring complex acts as a starting point or nucleation site for the growth of one microtubule. The centrosome as organizing center keeps the concentration of the free alpha-beta tubulin dimer low. In this way, cells can be regulated more surely where microtubules form. One microtubules are nucleated in the nucleation sites of the centrosome. It naturally grows outward from the organizing center for many minutes through adding alpha-beta tubulin dimers to its free end. After, it can suddenly undergo a transition that causes it to shrink quickly through losing tubulin dimers from its plus end. The microtubule may shrink partially and then quick starts growing again, or it may vanish entirely to be replaced by a new microtubule that grows from the same white tubulin ring complex. This extraordinary behavior of microtubule, that is, growing back and forth between polymerization and depolymerization, is known as dynamic instability. Such behavior is driven by GTP hydrolysis. Actin filament. Actin filaments are also known as microfilament, are another principal component of cytoskeleton. They are helical polymers of the protein actin. They are short, thin, and pliable structure measuring 7 nanometer in diameter, allow animal cells to adapt a variety of shapes and perform a variety of functions. They are built into a diversity of linear bundles, two-dimensional networks, and three-dimensional gels similar to microtubules. They have structural polarity, a plus end and minus end. They can be found throughout the cell. However, They are mostly highly condensed in the cortex, the layer of cytoplasm right below the plasma membrane. Actin filaments are made up of actin monomer, and they can grow by the addition of the monomers at either end, just like the microtubules. However, their growth is faster at the plus end than the minus end. A bare actin filament, like a microtubule without associated proteins, is characteristically unstable, and it can disassemble from both ends. In living cells, free actin monomers carry a tightly bound nucleoside triphosphate, in this case ATP. The actin monomer hydrolyzes its bound ATP to ADP soon after it is incorporated into the filament. So the function of actin filament is allow the animal cell to adapt a variety of shapes and perform a variety of functions. Next is the intermediate filament. Intermediate filament are another component of cytoskeleton that is responsible for defining the shape of cells and in the intracellular communication. They are rope-like fibers that measure 10 nanometer in diameter and approximately 7 nanometer, way smaller than the microtubules. They are made of fibrous filament proteins, strong yet tensile strength and durable. They are the toughest among the three components of cytoskeleton. They are of different types. One of types known as the nuclear lamina forms a meshwork just under the inner nuclear membrane. However, other types extend across the cytoplasm, providing cells mechanical strength and distributing the mechanical stresses in the epithelial tissue by spanning the cytoplasm from one cell junction to another. Though they deform, but they do not rupture. Given their unique properties, they do not directly participate in cell motility, unlike the microtubules and actin filament. Types of intermediate filament proteins. Intermediate filaments are made up of several types of proteins, having a general structural organization which are expressed in several types of cells. There are over 70 various subtypes of proteins that have been identified, and they are grouped into 5 types according to their matches flanked by their amino acid sequence, as you have seen in the table. Cell motility. Cell movement are made possible by the works of actin filaments in addition of myosin motors and the actin binding proteins, especially in the several active events on non-muscle cells. Examples of these active events include the following: cytokinesis, phagocytosis, cytoplasmic streaming, vesicle trafficking, blood platelet activation, cell locomotion, and changes in cell shape. Especially in cell locomotion are needed for many activities in higher vertebrates, including tissue and organ development, formation of blood vessels, development of axons, wound healing, and protection against infection. Cell locomotion are contribute to the spread of cancerous tumors. The following figure shows the cell locomotion over the substratum, although you can see that the movement is repetitive and it is similar to the movement as you literally walk, though motile cells assume different shape as they crawl over the substratum. However, they have similarities in the series of events, as you can see in the image below. Now for the last topic of this review, the cellular metabolism, the mitochondrion. It is specialized membrane-bound organelle where the exploitation of oxygen as a way to take out energy occurs. 2 layers of membrane cover a mitochondrion is the outer mitochondrial membrane, and the inner mitochondrial membrane. Generally, mitochondria generate ATP, the cell's energy currency, to proceed most of the cell's energy. They serve as primary supplier of ATP in non-photosynthetic tissue. The outer membrane is approximately 50% lipid by weight and holds a peculiar blend of enzymes involved in a diverse of physiological activities. The inner membrane forms several folds called cristae and extend in the matrix or the interior of the organelle. The matrix has a gel-like makeup and contains enzymes, minute ribosomes, and DNA molecules. This non-chromosomal DNA encodes a small number of mitochondrial polypeptides tightly incorporated into the inner mitochondrial membrane next to the polypeptides encoded by genes inside the nucleus. So what is the function of mitochondria? They serve as the primary suppliers of ATP in non-photosynthetic tissue. They serve as primary suppliers of ATP in non-photosynthetic tissues. It is a source of ATP in photosynthetic leaves during the light-independent reaction of photosynthesis. They function as the formation location of several substances encompassing specific amino acid and the heme group. They perform a critical function in the uptake and release of calcium ions. Together with the endoplasmic reticulum, they help regulate calcium ions concentration in the cytosol. They also play an essential role in the process of apoptosis or cell death. Aerobic respiration in the mitochondria means that such a metabolic process requires oxygen. However, the process of ATP generation takes place first in the cell cytosol, a process known as glycolysis. One of the products of glycolysis, called pyruvate, will then undergo oxidative phosphorylation before the next step of the process to proceed in the mitochondria, where the rest of ATP synthesis occur. The chloroplast. The chloroplast is a plant cell organelle known to perform photosynthesis. It contains the pigment that captures sunlight energy and converts it into chemical energy to make glucose. Three membranes divide the chloroplast into three distinct compartments. They include the following: the intermembrane space that is located between the two membranes of the chloroplast envelope, the stroma, which rests inside the envelope but outside the thylakoid membrane, and the thylakoid lumen. Generally, the chloroplast is responsible for generating ATP. Its function for the photosynthetic conversion of carbon dioxide to carbohydrates. Specifically, it synthesizes amino acids, fatty acids, and lipid components of their membranes. In addition, it helps reduce nitrite to ammonia, which is critical period in integrating nitrogen into organic compounds. In particular, the chloroplast envelope function to regulate the entry and exit of molecules. The outer membrane is permeable to ions and contains purines and facilitates the easy passage of smaller molecules. The inner membrane is impermeable that limits the movement of molecules between cytosol and the interior of the organelle. The thylakoid membrane functions for the electron transport chain and the generation of ATP. So let's talk about the process of photosynthesis. The photosynthetic process of plants involves two major reactions, the light-dependent reaction and the light-independent reaction. The former requires a light source for it to occur. However, the latter will proceed even without the presence of light energy. However, it will proceed using the first reaction products such as high energy storing compounds, the ATP and the NADH.